Hello, morning group. Oh, how's everybody doing? It's Wednesday. Who felt the earthquake last night? Only a couple of you, huh? That one woke me up. Usually I can sleep through them, but that one was like, Bacha! It was kind of like, I thought something ran into the house. That was a doozy. Cool. I think the Earth was just excited to celebrate Earth Day. So it was just like, woo, let's get the party started. Like, because it was like at 12:02, it was like, all right, it's Earth Day, let's do this. Okay, we'll jump right in. We'll work on another FRQ today. This is an FRQ from Unit 3. It is a, let me confirm before I lie. It is a short FRQ. So that means it's 15 minutes. It's definitely different than any of the short FRQs that we've done before. So that's good. It'll require you to do a little bit more describing or storytelling today, so that's good. So let's just flash it up on the screen, give you a couple minutes to copy it down. You guys see it okay? It's kind of small. It's okay, yeah? Most of you are nodding yes. Okay, I'll read it aloud as well. It says, the effects of pH and temperature were studied for an enzyme catalyzed reaction. The following results were obtained. And then you have two small graphs. And then it says, describe a controlled experiment that could have produced the data shown for either temperature or pH. Be sure to state the hypothesis that was tested here. I hope you guys are staying more organized than I am. I have papers just everywhere. It's incredible. I need just like a good old-fashioned real notebook. Like a line, spiral, you know, ones that you guys have. Alrighty. Alrighty. Okay. I can always put that back up if we need it again, but let's go ahead and just jot down some of our friendly tips and hints for today. So you can almost be guaranteed, actually I can guarantee you that you are going to have one question that shows you some kind of graph that you're going to have to interpret. Oops, sorry. Yeah, let's get it right the first time. So if you, I mean, for the most part, I think you guys have been reading graphs since you were pretty little. So at this point, I'm sure you can, read a basic graph, especially the ones that are in this problem. But just, getting more practice with that, getting more practice at looking at a table or a chart and starting to be able to visualize a graph before you even make one. Practice like that is going to be really helpful because the faster you can interpret graphs or tables, the more time you'll have to respond to them. So that's what's going to happen with this case, because you don't want to spend a ton of time staring at these graphs because you only have 15 minutes to respond. And in this particular response, they ask you one of our favoriteest AP terms, which is to describe. We've said this a bunch, but describe is to give an account of. Convey. Characterize. Right. This should all scream for you now that you're storytelling. So anytime they give you describe, you know that you have a few sentences ahead of you, which is different from like identify, which should be a one-sentence answer, right? Or predict, which is a hypothesis, that's also one sentence. But when you get the words like describe or discuss or what's another one? I guess those are the two big ones where you're going to write multiple sentences based on your response. Okay, so we're describing today. And what are we describing? We're going to describe a controlled experiment. And I just want to take a second to make sure we're all clear about what a controlled experiment is. Controlled experiment just means that you can measure some kind of variables or data. You can think of it as something that you set up. An uncontrolled experiment is one that's just, hey, I'm gonna look at this area of stuff and just notice. I'm going to take an account of what's going on in this environment, but not necessarily control it in any way. I'm not counting how many rabbits are there or how much rainfall was there. I'm not collecting any data. I'm not setting up any data. No, there's no amount of time set. Controlled experiments have numbers that go with it. So you really want to think of it as quantitative analysis. It's a tough one to spell, but I spell it right. Not to say that this excludes qualitative. But most experiments have numbers in them. Alrighty. Last but not least, you will have to write a hypothesis. And I'm sure this is a broken record for you, but a hypothesis is an if-then statement. If you want to go one step further, which is how I normally teach hypotheses, you're going to also add a because. Now the because part is kind of justifying it, justifying your reasoning. But I think it's important. Why not? Doesn't make it a run-on sentence. It doesn't, you're not adding anything you're not supposed to. So I always ask students to add the because. We did this one, we did a hypothesis in class yesterday. So you should be well versed in writing these. Okie dokie, I have 8:12 on the clock. So we'll go for about 10 minutes and I'll check in with you and see how you're doing. Again, describe, that's endless amount of sentences, but at the same time, you still want to be concise so that your reader doesn't have to guess what you're talking about. They don't just search for your answer. Be clear, tell a story, but tell it quickly. Go ahead. Before you get too deep into it, I just want to point out some of the wording. It says, describe a controlled experiment that could have produced the data shown, for either temperature or pH. So this can be a little confusing because you could think, oh, I just need to describe an experiment that talks about temperature or an experiment that talks about the pH. No, because it wants you just to describe one experiment, a controlled experiment that could produce the data for either of these. So meaning it's one experiment that encompasses both of these results. Okay, so it's not, I get to pick an experiment. It's one experiment that can give you both of these. So think about that when you're describing how your experiment will go.
Miss Masonette?
Yes.
Wouldn't that mean that there'd be two independent variables going on?
You mean as far as the temperature and the pH go?
Yeah, because if we're trying to describe an experiment that would encompass both.
Yeah.
Okay.
Yeah. Yeah.
Thank you.
You got it.
Wait, so we do it for both experiments or just one?
So it's one experiment that can test both of these, that can end up with these results, both of them.
Okay.
So it's basically like Chanel was saying, it's just adding an extra independent variable. You're testing two things at once. So one experiment gave both of these results. How?
It says either or.
Right, but it says to describe one experiment that could show the results of either of these.
Oh, okay.
Does that make sense? So within one experiment, you should be able to get the temperature graph or the pH graph. Which also means both, because one experiment took care of both of them. Okay, we're creeping up on 10 minutes here. Just checking in on you. Anyone need more time? Yep, okay, maybe a couple more minutes. How we doing? Good? Okay. All right, let's chat about this one. I'm going to just put it back up on the screen so we can see it, maybe. Yep, there it is. Okay, so this one was, a little bit more, what's the word, subjective, in that, like, all of your answers are going to vary depending on how you wanted to describe your experiment. And remember that describing means you can use multiple sentences to tell a story. So, there's definitely room for getting a little carried away here. You definitely wanted to be pretty concise when it comes to your experiment and a little bit vague even. You don't want to be like, first, take 10 milliliters of, amylase solution. Like, it's a little bit too, too precise when it comes to describing that experiment. So there is a way to get too descriptive here. Does anyone want to share some of their experiment? or the broad strokes of it? You don't have to read your answer if you don't want to, but just what your experiment might look like. Sadie, great, thanks.
So I did an experiment that would use enzymes that produce carbon dioxide bubbles.
Oh, cool. Oh, perfect.
So you would put the enzyme into a solution with substrates, and you would count the bubbles after 10 minutes at different pHs and temperatures.
Okay, so the let's say they're tubes or something. The tubes were put at varying temperatures and also varying pHs.
Yeah.
But the time signature was the same.
Mm-hmm.
Gotcha. Okay, cool. I'm just thinking about that. And you said you were going to count your bubbles. Yeah, that all checks out. That sounds good. Okay. Anyone else? I'm sure you all have different experiments. Chanel, go ahead.
I kind of did the same thing, but more like I went back to like a photosynthesis lab we did.
Okay.
And so I said we could put like a plant in an acidic type water so that when it produces oxygen gas, the water would become more basic. So that way it isn't like another independent variable.
Okay.
And then we could still see how enzyme activity goes.
Okay.
And then we would put it under different varying temperatures.
Gotcha. Okay, so the water itself is the pH indicator. Making, great, so that made your pH almost like a dependent variable, like a second dependent.
Yeah.
Great, so you had varying temperatures of environment. Your plant was doing enzymatic activity just in terms of cellular respiration, basically, like the whole kit and caboodle, doing photosynthesis, like you said. Great. And you're measuring then oxygen output as a measurement of your enzymatic activity. And you're measuring the pH after the fact. Does that sound right? Did I re-describe it to you? Perfect.
Well, actually, like because as pH would change, wouldn't the enzyme activity change as well?
Absolutely, yes. Yeah, so I mean,
It
really,
could still be independent.
Yep, and really you could go either way with this. Having answered your question earlier and then kind of thinking about it myself, there are two different ways you could spin this experiment where pH is an independent or a dependent variable. So yeah, there's definitely multiple ways to look at this one and still be right. The idea here is that everyone's being super clear for their reader about which is which. And the best way they can tell after they read your story of your experiment is how you talk about it in your hypothesis. Does that make sense? Because then they're clearly gonna be able to see, that if they say if, and then they name some independent variable, and then they name some dependent variable, they're gonna know which one you're talking about.
Okay.
Do you want to share your hypothesis just so we can kind of like test that theory?
Yeah. I said, and the hypothesis for this experiment is, if temperature is varied along with pH, then the highest enzyme activity will be at like 55 degrees Celsius or like 7 pH, kind of specific numbers.
Perfect. Great. So clearly for me as the reader, I'm hearing it that both your temperature and your pH are independent in this particular experiment.
Okay.
That's what you were hoping for?
Yeah.
Great. Perfect. And the only thing I would say is to, if you can, add a because at the end of that. If blah, 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 then blah, 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 because what? What is this whole experiment telling us? I mean, when you look at these graphs, they look very similar, right? And the independent variable in both of them, basically you're saying that those independent variables are acting similarly. So for everyone, your because of your hypothesis should be something like temperature and pH are directly related. When one goes up, the other goes up. When one comes down, the other comes down. Anyone else want to share their experiment or their hypothesis or both? Or
I have
questions?
a question.
Yeah, hello.
Do we have to provide like a specific temperature range or pH
Nope.
range?
Nope, absolutely not. You don't have to at all. I would say that if you do, that you know that you're obviously consistent, that you're not just picking like arbitrary numbers. Like Chanel's obviously not going to pick, 375 degrees Celsius, because what planet is she living on, right? So she wants to make sure that if she is going to pick numbers, she's going to pick numbers that make sense to, earthly organisms that we have learned about. But at the same time, being vague with your experiment can be really beneficial as well, where you're just saying, the tubes were placed at varying temperatures. No numbers necessarily indicated. You're just talking about what the experiment could do. Does that make sense, Sheila?
Yeah, I had a question. So, you know how you said that, like, it means like pH and temperature are related?
Yep.
Would we still be able to say that if pH and temperature are the independent variables? So they're supposed to be affecting like the enzyme activity. So like how would
Yes.
we say that like they are related if we're saying that the pH and temperature are the independent variables?
I see what you're asking. How do I answer that?
Is it that they're related in how they act, but they're not actually like?
Yeah, it's not necessarily that, we can't prove with our experiment, unless you wrote an experiment where you could, I'm not sure, but we couldn't prove that, if pH goes up, that the temperature of the environment would go up too, or vice versa. But in general, both of these are trending the same way. So maybe, they're definitely directly related because they trend the same, but it's just that those two particular variables of enzymatic activity tend to do the same, what's the word? Oh, they act the same. I don't know how else.
The same.
Yeah, they're just
like they can have the same effect almost, but not really on an enzyme.
Correct. That's exactly right. And in a similar trend at, that they both go up and then they come back down, that there's an optimal range for both of these things as far as enzymatic activity goes. So maybe I'm not saying it clearly, but the idea is that, at the end of your hypothesis, that you're able to say that both temperature and pH have a similar way of acting upon the enzyme activity. Is that helpful?
Yeah, but I kind of want my hypothesis a little differently.
Sure, you want to read it?
Like I said, like if an enzyme is placed in an environment with its optimal temperature and optimal pH, it will produce the most product because it's in an environment where it can work optimally and lower the activation energy as low as possible to catalyze a reaction.
Oh yeah, I like that. That's great. Yeah, so you took it and you made it more of a broad hypothesis for enzymes in general and activation energy, which is great. So yeah, that's a perfect hypothesis. Do you mind telling me about your experiment?
Sure. I basically said that like a scientist placed an enzyme like and its substrate in different environments with different temperatures and pH values. And I said that the scientist would test the enzyme activity by the amount of product that the enzyme produced in each different environment. Great.
And then I would just maybe say something, because you come back to the word optimal temperature and pH as an indicator of low activation energy. Maybe just say something in your experiment that the experimenter was looking for, low activation energy, high output, something like that. That was the point of their experiment.
Yeah.
Just so that when you circle back to your hypothesis, or like they assume that if you wrote the hypothesis first, it makes sense for your experiment.
All right.
Right, because, and this goes for everyone, when you're, in theory, if we're being good scientists, right, we write a hypothesis first and then make up an experiment that can prove that hypothesis. or at least test it. So even though in this case you probably wrote your experiment first and then wrote your hypothesis based on that, which is fine, we just want to make sure that they are related to each other, that whatever you wrote for your hypothesis was definitely clearly tested in your experiment. That's what they're looking for here. They're not necessarily looking for in this question, you know, how much you know about enzymes or how much you know about temperature and pH. They want to know that you can design an experiment here based on a hypothesis that clearly has independent and dependent variables. Questions or anyone else want to share? Okay, well, per usual, if you'd like, you can send me your FRQ and I'm happy to give you some feedback. Lots of you have been doing that and I hope that you find it helpful. In addition, what else was I gonna say? Some of you I will see on Thursday in class. Some of you I already saw yesterday in class. You've got your study guides to watch this week and a couple FRQs to do. I'm just kind of talking to myself right now, make sure there's no announcements that I have. Today's Earth Day. There's, there was supposed to be this huge eco festival today. I don't know if you had heard about it. And it's called the South Bay Eco Festival for Earth Day. And they're doing everything virtually online today. So if you are bored and you want to check out the Earth Day activities, southbayecofestival.com, I think, or something like that. That'll be really fun. I guess that's it, right? I don't know. It's gonna be gorgeous weather the rest of the week. Get outside, enjoy some vitamin D, and hopefully I'll see you tomorrow. All right, guys, have a great day.
Thank you.
Bye.